Hello, everyone. Welcome to our next talk, and I'm very happy to announce our next speaker, Jane Mellor. So I would like to start with saying a few sentences about Jane's career path. During her PhD, Jane was working with Fred Brown and Tim Harris on the translation of foot and mouth disease virus at the Animal Virus Research Institute in Pdgfra, UK, and has successfully completed her PhD in 1982. Afterwards, Jane has moved to the University of Oxford, a Department of Biochemistry, for her postdoctoral research with Alan and Sue Kingsman, where she was focusing on gene expression system in yeast. In 1989, Jane became the Wellcome Trust Senior Research Fellow, and in 1995 received a professorship. So Jane's group, established the Department of Biochemistry in Oxford, focuses on gene expression and chromatin. And her group has pioneered in understanding the role of non-coding transcription, demonstrating the roles in switching chromosome conformation, signatures to coordinate gene expression with metabolism. So, of course, we can't wait to hear about this topic from Jane herself. So, Jane, please, the stage is yours.
Okay, well, thank you very much. It's a real pity that we can't meet in person today, but I'll do my best to give you a little vignette of the sort of questions we're asking to deconstruct the relationship between chromatin, metabolism, and gene expression. I always like to start my talks by acknowledging the people who've done the work today. And the work today has been done mainly by Sylvia Ranieri and Jack Feltham, supported by the other members of my group. Our collaborators, Oscar Yanes, who has helped us with metabolomics and metabolic flux analysis, Scamantis' group in Oxford, who've done TAP sequencing to measure levels of DNA methylation. Andrew Angel helps us with modeling, Andre Ferger with gene expression, Roman Benedict, Sabrina, and Shabazz, who help us with proteomics analysis. And of course, I can't not acknowledge our funding bodies who have funded the work I'm going to talk about today. So one of the things we want to test is the hypothesis that there's a linear flow of information from the chromatin, so chromosome conformations and epigenetic modifications on the chromatin, to transcription, RNA, translation to protein, those proteins then affecting pathways, metabolites, post-translational modifications to proteins and modifications to lipids and DNA, which then gives us the phenotype. And there's quite a lot of data in the literature which supports these sorts of hypotheses. But what I'm hoping to show you today is that it might not be quite so straightforward. And what I'm going to do is use our experimental analysis model forward by Brad Bernstein's group, which was published in this seminal Nature paper in 2017, which led to the idea of this relationship between chromosome conformation and phenotype using a disease model. I should also say at this point that we've been using other systems to test this linear flow model, and we have some data which is currently in second review, which is available on bioarchives using yeast to test the linear nature of gene expression. And what I hope to convince you by the end of this talk, and of course you're welcome to challenge me in the question session, is that there is not really a linear flow of information from the chromatin to the phenotype, and it's much more likely to be a branched pathway. But anyway, the system I'm going to use to deconstruct this model is a system defined by the Bernstein group. And they were looking at disease-associated mutations, and the disease they were looking at is glioma or glioblastoma. And they were looking at mutations which specifically lead to a change in metabolism that they proposed altered the chromatin structure. And the chromatin structure they were looking at are domains in the chromatin. So these are the higher order structures, the way the chromosomes and chromatin folds, which are known as topologically associated domains. And they proposed a model whereby disease mutations associated with glioma alter the way these TADs fold, and this then leads to changes in gene expression to altered phenotype and to the disease. And very specifically, their model suggested that changes to the levels of DNA methylation altered the ability of this protein here called CTCF to form an insulator, which would normally stop this strong enhancer talking to the promoter of this oncogene Pdgfra, and that when this structure is lost, the enhancer activates the oncogene, the platelet-derived growth factor receptor alpha gene, and it is this that leads to the brain cancer. So that's the hypothesis I want to deconstruct with you today. So let me just introduce a little bit more about how we think this works. So here is the Pdgfra gene here. This is the putative insulator defined by the Bernstein group. This is the DNA binding sequence to which CTCF binds, and you'll notice in the middle of it there's a CG dinucleotide. And those of you who know something about epigenetics will know that CG dinucleotides can be subject to methylation at the 5' position of the cytosine. And many DNA binding proteins, particularly CTCF, are sensitive to the levels of DNA methylation. And their model was that changes in levels of DNA methylation affect the ability of this enhancer here, which normally activates this FIP1L1 gene here, which is a strong enhancer and a strongly expressed gene. This now allows this enhancer to talk to the Pdgfra promoter, which is down here, and lead to a 5-fold increase in Pdgfra expression, as I said. So what this does is relates a change in metabolism to a change in DNA methylation to a change in chromatin organization to changes in gene expression. So let's just deconstruct this a little bit more. So changes to DNA methylation prevent CTCF binding and leads to loss of this insulator, which would normally separate these two genes and this enhancer. And when we lose this insulator, the FIP1L1 enhancer interacts and activates expression of the promoter from the oncogene, leading to the development of disease. So the question is, how does this happen? So that's the first question I'm going to address with the how question. Well, we know that a lot of cancers, particularly gliomas and glioblastomas, are associated with mutations in a gene encoding isocitrate dehydrogenase. So IDH mutations. And this is true of gliomas and other cancers such as AML. So it's quite a common mutation in cancers. So about 80% of the grade 2 and grade 3 gliomas and astrocytomas are associated with IDH mutations. So how does this work? So this is the model. So to propose the model, the Bernstein group, Flavahan in particular, started off with biopsies from patients who presented with glioblastoma. And they divided them into two classes. Those who had a specific mutation in their IDH genes. This is an arginine 132 to histidine mutation, which is a gain of function mutation. And I'll come back to this in a minute. So they stratified them into two groups, so with the mutation or without the mutation, but both patients presented with glioblastoma. And what they observed is that the patients who had the mutation showed at the cohort level, so when they put everybody together, generally showed an increase in Pdgfra expression and more concordance with the levels of FIP1L1 expression than those with glioblastoma but no IDH mutation. In other words, this would normally be highly expressed, this would normally be poorly expressed, but in these particular patients, this was more highly expressed, so they were seeing more concordance. And actually, they discovered this locus to study by doing a study bioinformatically. So the basis of this study is pure bioinformatics. So effectively what they did was they took high-C maps, which define topologically associated domains from a wide range of different tissues, and looked for boundary elements, and then took the genes that were present in different topologically associated domains and looked in disease states in the patients with the mutation or the patients without mutation, a change in the concordance between the levels of expression of, in this case, FIP1L1 and Pdgfra. But actually, in their initial analysis, they defined 203 different sites in the genome where patterns of gene expression changed, consistent with losing an insulator and a gene increasing expression because it would come under the control of an ectopic enhancer, which would normally be present in another gene. And they then went on to do 1 or 2 experiments to sort of validate the model, but they never actually did what you might call a full validation. So they never actually showed changes in the higher order structures, or that this was a function of this enhancer or methylation at this site. So there was a lot of unanswered questions in this model that we set out to address. So just to recap, insulators that separate topologically associated domains can block the interactions between enhancers and promoters and therefore control gene expression. So when we have a functional enhancer, it generally binds this protein CTCF, or there are other insulator binding proteins, but CTCF is a common one. And CTCF binding is DNA methylation sensitive. So when the C at the 5 position is methylated, CTCF can't bind. And then the enhancer can talk to the promoter and the gene is switched on or upregulated. So that's the first thing to note. The second thing to note is that levels of DNA, and this also applies to protein methylation, so modifications by adding methyl groups, these are influenced by the concentration of central metabolic intermediates. So if we look at DNA methylation, this specific example here, here's our CG dinucleotide, and you can see that it's symmetrical. So in the presence of S-adenosyl methionine, which is our central metabolite, which is the methyl donor, the DNA methyl transferases will deposit methylation symmetrically on the 5 position of the Cs. And that in the case of an insulator is sufficient to prevent CTCF binding. But there are a second group of enzymes called Tet enzymes, which stands for 10-11 translocation methyl cytosine dioxygenases, which are able to demethylate cytosine, so they remove the methyl group. And they do it first by converting 5-methyl-C to 5-hydroxy-methyl-C, and then the hydroxy-methyl-C is removed to demethylate these residues. Now, these enzymes require, as their cofactor, another central metabolite, alpha-ketoglutarate. So some of you will, if you cast your minds back to your first year biochemistry course, will remember that alpha-ketoglutarate is an intermediate on the TCA cycle, and isocitrate is converted to alpha-ketoglutarate by the isocitrate dehydrogenase enzymes. And remember, these are the ones that are mutated in our model system here. This is just to remind you that there is also quite a complicated set of central metabolic pathways which control the production of S-adenosyl methionine, which is our methyl donor. But we're really interested in this side of the pathway, the demethylation pathway, which is alpha-ketoglutarate dependent. I should say at this point that there are 3 isocitrate dehydrogenase enzymes in mammalian cells, 2 in the mitochondria and 1 in the cytoplasm. We are actually going to be dealing with IDH1, which is a mutation in the cytoplasm, but both forms of the enzyme, wherever they're located, will convert isocitrate to alpha-ketoglutarate. And the alpha-ketoglutarate is removed from the cytoplasm into the nucleus to provide the cofactor for the Tet enzymes to demethylate DNA and demethylate protein. So I described in the Flavahan model a mutation, the R132H mutation in IDH1, which is an activating mutation. And this mutation here, and where I show it, I show the asterisk to show it's an activated mutation. So in this gain of function mutation here, the enzyme becomes overactive and converts the alpha-ketoglutarate to this Onco metabolite. So in other words, a metabolite which is associated with cancer called D2-hydroxyglutarate. And D2-hydroxyglutarate is produced as a result of activating mutations, both using the mitochondrial and the cytoplasmic alpha-ketoglutarate. Now, what does 2-hydroxyglutarate do? Well, 2-hydroxyglutarate acts as a competitive inhibitor of the Tet enzymes. So in other words, it prevents DNA and protein demethylation. And the net result of having high levels of 2-hydroxyglutarate is that levels of DNA methylation and protein methylation increase. And by the model that I've shown you, this should reduce in loss of insulator function, particularly at the Pdgfra insulator, by increasing methylation levels and preventing CTCF binding. So here's the metabolism here. Here's our activated isocitrate dehydrogenase. Here's our 2-hydroxyglutarate acting as a competitive inhibitor of our Tet enzymes, which demethylate the DNA and also the protein. So alpha-ketoglutarate activates these enzymes, whereas this very highly structurally related metabolite inhibits them. I should also say, interestingly, that succinate and fumarate, which are the next two metabolites on the TCA cycle, also act as competitive inhibitors. So loss of function mutations in succinate dehydrogenase or fumarate hydratase will result in accumulation of levels of fumarate and succinate, and they can also inhibit this pathway and alter the function of insulators, and therefore presumably affect higher order structures in the chromosome. So many forms, as I've said, of glioblastoma and other cancers have these IDH gain of function mutations. So it's quite a common change in cancers. So what we did was set out to test this hypothesis by specifically addressing the question as to whether this is a linear pathway using the glioma model system. So just to remind you, disease-associated mutations in IDH, the R132H mutation, lead to increase in protein and DNA methylation due to the production of the Onco metabolite 2D or D2-hydroxyglutarate. And in this particular model, the idea is it leads to defective insulator function here as a result of increased DNA methylation, which causes loss of CTCF binding and reorders the higher order chromatin structures to allow activation of Pdgfra. So that's our model. Okay, so how do we test this? So to do this, we used the best system we could possibly find. And this was a glioma-derived cell line called U87. And we created an isogenic derivative which contained a heterozygous activating R132H mutation, which was engineered into one of the two IDH1 alleles. Now, the key to getting the system to work was having a specific inhibitor of the activated IDH1 enzyme, which is known as GSK864. So GlaxoSmithKline were looking for drugs to treat glioma with, and they reasoned that a good target for a drug would be this activated form of IDH. So we have a specific inhibitor. And we took a multi-omics genome-wide experimental approach to assay each aspect of the model separately. Whoops, sorry. Okay, so the idea is that if the linear hypothesis is correct, then inhibiting this activated form of IDH1 should lead to a reduction in the levels of the 2-hydroxyglutarate, and this should change DNA and protein methylation levels and the associated change in TAD structure in the chromatin and Pdgfra expression. So if they're all linked together, if you need the change in the chromatin to get the change in gene expression, by inhibiting one, you should affect the other, and this will allow us to test the linear pathway. So this is what we did. We had the two cells and we treated them with or without the inhibitor for 3 time points, 48, 72, or 1 week. Each experiment was done in triplicate, and every experiment had spiking controls, so we could do good controls. We set very high statistical significance levels for changes to make sure that we were looking at things which were not noise but real. And we measured a number of different parameters. I'm not going to show you all these results. I'm just going to pick on a couple to show you the bottom line. So we measured metabolic flux and metabolomics. So we measured the levels of various metabolites in these cells. We measured DNA methylation using TAPS. We measured CTCF binding using ChIP-seq. We measured some chromatin modifications, so lysine 27 acetylation and lysine 9 trimethylation using ChIP-seq. RNA-seq coupled with RT-PCR, qRT-PCR for validation, and 3C for chromosome conformation analysis. I'm not going to go through those data. So that's what we did, and we're testing this hypothesis. So this is the first result. So we added GSK864, and we were able to show that it does indeed inhibit the activated IDH1 and reduces 2-hydroxyglutarate levels and restores gene expression. So you can see here our metabolomics. So this is in the wild-type cells. There's virtually no detectable 2-hydroxyglutarate in these cells because all the alpha-ketoglutarate goes around to succinate and fumarate in the TCA cycle. But in our IDH mutant cells, this is just a background in the assay. You can see we see in 10 different experiments high levels of 2-hydroxyglutarate. And importantly, when we add the GSK inhibitor in this experiment for 72 hours, but this works equivalently at 48 hours, you can see that the 2-hydroxyglutarate levels are reduced way down to near wild-type levels. So we can change the level of our oncometabolite using the drug. We can change levels of gene expression. So here is Pdgfra mRNA levels. This is from the genome-wide analysis spiking control from 3 repeats. You can see that levels of the Pdgfra transcript in the wild-type cells are low and don't change when we add GSK to the cells. In the mutant cells, where we have the high levels of 2-hydroxyglutarate, we get, in this case, about a 4-fold increase in expression of the transcript, and we see an equivalent expression of the protein by measuring Pdgfra receptor alpha levels on the surface of cells. So this is translated into protein. And importantly, when we add the inhibitor, these levels are restored back to wild type. So we're getting a nice correlation between the levels of 2-hydroxyglutarate and expression of Pdgfra. Because we've done a genome-wide analysis, we can look in a little bit more detail about other pathways which are affected by the activating mutation in the oncometabolite and how these change when we treat with GSK. And you can see that there are changes, more broad changes in gene expression, as you would expect, because 203 loci were proposed by Flavahan to be affected by this model. And interestingly, we see a cluster of genes related to differentiation and development, suggesting that these cells are becoming a little bit more differentiated and a little less cancer-like when we add the inhibitor to them. So the conclusion from this experiment is that at this level, reducing 2-dehydroxyglutarate levels alters patterns of gene expression, including levels of Pdgfra. So the question is how, and the obvious thing we're going to test is the chromatin, because that's what the Bernstein paper suggests is linked to the changes in gene expression. So I'm going to show you first of all the data for CTCF binding. So the take home from this is the epigenetic landscape changes in the IDH mutant cells, but importantly, this is not responsive to GSK864 treatment. So the number of peaks of CTCF binding decreases in the IDH1 mutant cell. This is expected. We've seen this from data from other groups, and the model suggests that CTCF binding should decrease because we'll see a concomitant increase in the levels of DNA methylation by this model. So we see mainly a decrease in levels. There are a few loci where we see an increase. But the important result here is that when we treat these cells with GSK, and this is at times when we know that the 2-hydroxyglutarate levels and gene expression are restored to wild type, we do not alter the number of peaks of CTCF. We also know that DNA methylation levels increase genome-wide, consistent with there being many more CG sites in the DNA being methylated in the mutant cells. But again, our preliminary data suggests that these do not change on GSK864 treatment, again, when we see changes in gene expression. So the first 2 epigenetic marks are not really changing. And interestingly, when we focus in on the original insulator, which was originally proposed by this bioinformatic analysis to be something that might be sensitive to changes in DNA methylation at this site here, leading to altered CTCF binding, this enhancer talking to this gene. If you have a look at our data here, and this is 2 different time points, you can see in the wild type cells and in the mutant cells, we're not certainly seeing a significant change in the levels of CTCF binding. And importantly, neither in the wild type nor in the mutants is this affected by the GSK864 treatment, which should theoretically reduce the levels of the oncometabolite. So there appears to be no specific change in CTCF binding in the IDH mutants before or after treatment with a GSK at this bioinformatically annotated insulator. Now, the other interesting thing that suggests that there may be a lot more going on than was originally defined is if we map the topologically associated domains. So each of these blue lines represents the beginning and end of a TAD. So these would be the insulator regions here. You can see that we don't see an insulated region here in astrocytes, which are the brain tissue in which this TAD analysis has been done, and which is most closely related to the glioma type cells that you see associated with the brain cancers. Now, this could be an insulator which creates what is called a sub-TAD, so it could be a subdivision, but it's certainly not creating the overall TAD structure. And note, interestingly, that Pdgfra, this gene which is overactivated, occurs in a TAD boundary here, and is actually in the same TAD as this FIP1L1 enhancer. So it suggests that the bioinformatically devised model can't quite be the whole answer, either the whole problem or the whole answer to the problem. Okay, so if I summarize this part, we don't see any change in CTCF binding at the putative Pdgfra insulator defined bioinformatically by Flavahan in this IDH mutant, and no effect of GSK864 treatment at the concentrations and time points tested on either CTCF binding or DNA methylation, despite the fact that these concentrations and times do change gene expression. So this suggests that the linear model is not correct, and that changes in higher order chromatin structure are not required to change expression of Pdgfra as a result of the mutation and the oncometabolite. But nonetheless, the expression is still changing, but it's unrelated to the chromatin. So we've looked at a number of other modifications. I've mentioned lysine 27 acetylation. I just want to show you the lysine 9 trimethylation changes, because many of you may know that lysine 9 trimethylation is associated with heterochromatic regions on chromosomes, things like telomeres, centromeres, and blocks of heterochromatin in which you often find transposons and things. So we wanted to have a look at this. And the model suggested that levels of lysine 9 trimethylation, which is a protein methylation, but demethylated by Tet enzymes, well, in some cases, should also increase. And in fact, if we look at our genome-wide data, we see 6,739 peaks, which highly significantly increase. But interestingly, at the same time, we see quite a lot of peaks which decrease, 2,660 peaks genome-wide in our IDH mutant cells compared to the wild type. You'll note yet again that treatment with the GSK inhibitor has no effect on these changed peaks. So just like with the CTCF binding and the DNA methylation, we're not seeing changes. So we expect to see increases in lysine 9, but I just really want to share with you the interesting data that we've got on the decreases in case anybody would like to pull this up. So the peaks both increase and decrease. So red are increases, decreases are blue in these mutant cells. Now, you can see that increases and decreases are present on all the chromosomes, but particularly the X chromosome shows a very large decrease in peaks. And as I said, the peaks that decrease are associated with blocks of lysine 9 trimethylation. You can see one of them here. You can see it scattered over the X chromosome here, which are these heterochromatic regions. You'll notice that in the mutant cells, which is this pink track here, lysine 9 methylation around the centromere, which is here, doesn't change at all. So the centromeric lysine 9 trimethylation levels are not changed, but many of these blocks on the arms of the chromosomes are reduced quite considerably. So, you know, if we didn't have the centromeric signal, you might say our chip hadn't worked, but we know that it has worked. Again, no responsiveness to the inhibitor in either the wild type or the mutant cells. And these blue lines represent our significant differentially decreased peaks. So we see no correlation between the changes in lysine 9 and protein coding gene expression or expression of the genes or the genetic elements like transposons, lines, signs, alloelements, which are a form of signs, which are often embedded and silenced in this lysine 9 methylation. So we see no changes in gene expression, but quite a dramatic change in the overall levels of lysine 9. And we're doing a microscopic analysis of this to see whether there is a change in the conformation of this chromosome in cells. I should also say we see this at other chromosomes, but to a lesser extent. So chromosome 6 and chromosome 18, just with the X chromosome, you can see that the centromeric lysine 9 trimethylation levels don't change, but we see changes in differentially expressed loss and gain. I mean, the gain peaks tend to be more single, and therefore they're harder to see on this chromosome-wide scale, but changes in these blocks of chromatin, which is quite interesting. So let me summarize. So while we can correlate, while we can correct the oncometabolic defect and the associated induction of gene expression changes, the GSK864 treatment at the concentrations and time point tested does not rescue any of the epigenetic alterations that we see induced by the IDH1R132H mutation. Gene expression is responsive to changes in metabolites, so the 2HG levels are reduced to wild type levels, but the epigenome doesn't change. It seems to be much less responsive to changes in metabolite concentration and is not directly related to changes in gene expression. So our conclusion is this linear model hypothesis is not correct, that the changes in the chromosome conformation are separate from the changes in gene expression, which do seem to correlate with the change in phenotype. So this is relatively preliminary data. It's very robust data, but we haven't finished the story. So there's a number of outstanding questions that we need to address, and I'd be very happy to discuss some of these ideas with you in the question session. So the first question is, how do oncometabolites affect gene expression? So they're affecting gene expression without, well, reduced levels of oncometabolites can affect gene expression without bringing about changes in the chromatin, which is normally what we would expect might change levels of gene expression. So one thing that we're looking at is that they do this by altering post-translational modifications, methylations, and acetylations, which I haven't shown you data for today, on transcription factors. And we know that transcription factors, their interactions with the DNA, certainly their binding kinetics and their turnover rates are much faster than you would see in the chromatin. So we are currently using our yeast model system testing the idea that it's post-translational modifications that are affected by changes in oncometabolite concentrations, and this can activate or deactivate PTMs via its, in this case, 2-hydroxyglutarate action on the demethylase and change the activity of our transcription factors, and these control gene expression. So the other question is, why do the chromatin changes not reflect changes in gene expression? Well, one possibility that we've been working on for many years is that these chromatin changes are simply not instructive for gene expression, as is widely believed. Now, for one of the modifications that we've worked on for a long time, which is trimethylation of lysine 4, which is a pretty universal mark present at the 5' region of genes which have the potential to be activated or are being expressed in eukaryotes from yeast to man, this is not instructive for gene expression. You can take away all this modification and gene expression doesn't change. So that's one possibility, that there's a strong correlation, but there's no causality between these marks and gene expression. And our current hypothesis is that these modifications reflect changes in gene expression. And this might reflect the fact that they change more slowly over time. So you change your gene expression, and then the modifications change to reflect the changes in gene expression. And they may help maintain a particular state, but they're not instructive for that state. And because we know that the chromatin and the DNA methylation marks change more slowly over time, this might explain this. So one take-home message from this talk is that you really have to take care when formulating a hypothesis based on bioinformatic analysis, where you're using correlations of data from different sources, as was done by Flavahan. So cell line, patient data, topologically associated domain data from many different cell lines, which came together to produce a very beautiful and very convincing story, but which looks like it's not quite right because it's correlations, but there's no causality. So it suggests that changes in higher order structure and changes in gene expression are reflecting something to do with the same change in the cells, but they're not causal. You don't have to have one changing in order to change the other. So one of the final points is that in 2007, when we started working, well, we started working on chromatin structure in 2000, but we set up a company called Oxford Biodynamics to exploit the ability of chromosome conformations to define the phenotype of cells, and humans come to that. So this could be, for example, in a human, the presence or absence of a disease, the likelihood that a person will develop a disease, or perhaps the responsiveness to a particular treatment regime, which is very important in clinical trials. But we know that changes in chromatin can reflect phenotype as well. But one of the things that we know from this long-standing data that we have on chromosome conformations is that these chromosome conformation changes are not always associated with changes in gene expression. So I'll leave you with this idea that this linear flow model, we think, based on our deconstruction of the Flavahan model here, which is shown here, and also from our yeast data here, which I said is available on bioRxiv, and I'll leave this slide up so that if anyone wants to get this URL down, you can, suggest that this is not a linear flow. And the current models we're developing is to put a focus on metabolites in the cell, because metabolites, although some metabolite changes will be responsive to changes in gene expression, we know that metabolite levels can change quite independently of gene expression. And it's these that we think are the key determinant of what's going on in the cell. So changes to metabolites, perhaps by changing post-translational modifications to proteins, for example, transcription factors, or changing the ability to modify DNA or lipids, as I said earlier, can lead to changes in phenotype here. And this could be, for example, through changes in gene expression. We also think that metabolites, again, perhaps through post-translational modifications, perhaps through the production of non-coding RNAs, which also change post-translational modifications to the chromatin, change chromosome conformations. But that these two events, the gene expression and the chromosome conformation, may be temporally quite separate, so they might occur on different timescales, but they're both reflecting changes in the cell at the level of metabolites. But what effectively you end up with is a chromosome conformation which correlates very strongly with phenotype, but doesn't necessarily require this branch of the pathway to produce that relationship. So an altered chromosome conformation can be associated with a new phenotype without a linear pathway of gene expression changes. And the idea is that these gene expression changes are occurring at a different time scale. And with that idea, I'll stop, and I'm very happy to take questions.
Great. Thank you, Jane. Very interesting talk. Let's start with questions. So Daniel asks, Usually cancer cells show global hypermethylation similar to stem cells. So is the observation in the glioma sample a locus-specific hypermethylation or also seen on the global genome level?
No, it's very specific. I mean, in the Flavahan analysis, they defined 203 loci, which they proposed based on the disconnect between levels of gene expression. That would be a limited number of loci which would change. Our DNA methylation data also supports increased DNA methylation in these tumor cells. The gliomas also show a GIMP phenotype. So promoters also become hypermethylated, despite there being hypomethylation generally. And this can also alter patterns of gene expression because promoter methylation is often associated with downregulation. So it's a complicated story in these cancer cells and cell types. That's all I can say at the moment. But there is definitely, and other people have observed at Turcan's lab who have worked extensively on gliomas for many years, observe the GIMP phenotype, and they haven't focused in that lab on insulators, but at certain insulators we do see changes in DNA methylation, although not at this particular one that was defined bioinformatically. Does that answer the question?
Yes, I think so.
There's no comeback here, so it's very difficult to do questions.
Yeah. The next question is from Julio.
Mm-hmm.
What other metabolites that cause changes in gene expression are you currently studying?
Okay, so the other one we're currently studying is acetyl-CoA. Now, interestingly, acetyl-CoA is associated with acetylation of histones and acetylation of proteins. Many transcription factors are both acetylated, methylated, and phosphorylated. To a certain extent, we're looking at phosphorylation as well, but only through changes in ATP levels, which reflect, for example, changes in mitochondrial activity in the context of yeast. Now, interestingly, in these activated glioblastoma cells with the IDH-R132H mutation, although the 2-hydroxyglutarate levels change like 200-fold, I mean, there's a massive change, massive upregulation, which can be downregulated by this specific inhibitor. We do also see changes in the levels of other metabolites, including acetyl-CoA. And interestingly, these changes are not sensitive to the GSK inhibitor. So there's something about resetting the chromatin in this long-term glioma model, which is more generally affecting metabolites and affecting things like, as I said, levels of acetyl-CoA, which go up about 2-fold. And we know that one of the consequences of this is increased acetylation of the histones and increased acetylation of other proteins. And this is the current area that we're studying. With a 2-fold change, it's quite challenging to study this sort of change in the mammalian cells, but we are studying changes in acetyl-CoA levels in yeast in a system where the changes are much more dramatic over time in a synchronized population. And we can study temporal correlations between changes in acetyl-CoA and ATP, changes in the chromatin and changes in gene expression, and begin to ask, just as we see in the mammalian cells, whether these are correlations or causal relationships. And our current idea is that they are correlations again. So there's a disconnect between what's happening at the chromatin and gene expression. It also means that we need to get our proteomics analysis up to a level which is challenging for even the best proteomics labs in the world, where we can measure post-translational modifications on transcription factors globally. This is just measuring transcription factors, which are often very lowly abundant, is challenging in proteomics. And then combining that with changes in post-translational modifications is really pushing the technology. This is the way we're trying to go, because we believe that, as I said, that it's the PTMs, the post-translational modifications, acetylation, methylation, phosphorylation on the transcription factors, which is responding to the changes in metabolism. So the answer is acetyl-CoA is the one we're really focusing on.
Great, thank you. Next question is from Mrna.
From who? Sorry?
Mrna Nolt.
Okay.
Actually, she has two questions. I will read the first one first. Chronic stress is known to impact mitochondrial functioning. Can you hear me properly?
I can hear you, yeah.
Chronic stress is known to impact mitochondrial functioning, while neurons mainly perform oxidative phosphorylation, as opposed to glycolysis in astrocytes. Could impairment in energy metabolism due to stress be causal for higher incidence of cancer in chronically stressed individuals?
That's perfectly possible. We are not studying that, but we're very interested in these two different forms of metabolism. So the glycolytic-based fermentative type, low oxygen, versus the mitochondrial metabolism. And we're using a yeast system called the yeast metabolic cycle, which naturally cycles between these two different stages of being primarily glycolytic and then primarily oxidative, which is associated with high metabolic activity, high use of oxygen, high levels of ATP, and high levels of things like acetyl-CoA, and trying to understand how different genes get switched on and off in those two conditions. So I think this is a really exciting area for future research. As I said, we're not looking at this at the level of cancer cells, we're looking at it at the level of a yeast, because we have this beautiful system where the yeast naturally change between these two different levels without any sort of intervention. The problem with studying cancers is that you have so many other underlying changes, and even in the system we're using here, where we're trying to make the change in this system as isogenic as possible, we know that once cells have been in culture for a while, they accumulate all sorts of secondary mutations, and they can never be described really as isogenic. But I think for the future, studying cancers with different primary metabolic pathways and trying to relate how stress relates to metabolic function in terms of mitochondria is a really interesting area for future research. I'm not doing that, but if anybody wants to go in there and really define really important new pathways, especially related to the way humans live now, I think that's really going to be really informative and should generate a lot of really interesting data.
Great. I think I'm going to read the last question because we're a bit behind the schedule. So This question is from Tu.
Yeah.
It's a very interesting. Thanks you for your interest and talk. And the question is, would you think introducing spatial temporal control in the study could help in clarifying the relationships?
Absolutely. Absolutely. I mean, we've tried to do this, with the at the global level on a cell population using our inhibitor, but I think we really need to go in and do some experiments on single cells where we can define exactly what stage a particular cell is at when we add the drug, what happens, how long it takes for things to happen at an individual cell level. We're going to have to do it on a population of cells, but we'd really like to get to the single cell level where we can introduce these spatial temporal controls. I mentioned one of the things that we're doing with these lysine 9 methylation changes that we see, where we lose our blocks of heterochromatin, is we're using SIM, which is structural illuminated microscopy, which allows you, together with light sheet, to get really detailed views of what's happening to the chromatin, to see whether the heterochromatin is changing in these cells, because of course we could be having consequences of where things are in cells, which influence how things change, as opposed to actually what's going on at particular loci. So that's a really interesting area, and I think one, again, ripe for exploitation, for exciting new data. We haven't got any data yet. We've done the first experiment. I've got some images, but we haven't got the detailed analysis yet. It's looking interesting. That's all I'm going to say.
Great. Thank you so much, Jane, first of all, for your time and also for a very interesting talk.
Okay.
And I would like to thank again the attendees for interesting questions. And I have a few small updates. So first of all, our next event is poster session number 3, which is happening at 3:30 German time in 15 minutes. And please vote for your favorite poster afterwards. And I would like to remind that we have a social event today at 8 p.m. German time, which is called Horizon Sunrise. So please join us. And thank you all again. It was nice having you here. And see you on the next talk. Thank you, Jane.
Thank you. And thank you, the audience, for listening and for the great questions as well.